Welcome back to our channel, where we are deeply committed to uncovering the latest in medical breakthroughs and drug development. Today, we continue our exploration of AOH1996, an experimental drug that's been stirring excitement across the medical community for its broad potential in cancer treatment. The versatility of AOH1996, showing promise across various tumor types, has captivated our attention. In our quest for comprehensive coverage, I stumbled upon an enlightening article on Wikipedia, authored by a renowned expert in the field. This discovery has fueled our latest video, aimed at providing our subscribers with the most up-to-date and thorough analysis of AOH 1996 and other significant medical trials. We're here to dissect complex medical jargon and present you with clear, accurate, and compassionate insights into these life-changing developments. Creating this content demands extensive research, digital iteration, and a relentless pursuit of perfection to ensure the information we share is both accurate and impactful. Your support is crucial to our mission. By subscribing, liking, and sharing our videos, you not only stay informed about groundbreaking medical advancements, but also help us reach and educate more people. Engaging with our content through comments also greatly assists in enhancing our visibility and supports the extensive work involved in producing these detailed videos. So join us on this journey of discovery and enlightenment. Let's dive into the world of AOH 1996 and beyond as we unravel the mysteries of medical science together, fostering hope and understanding in the complex world of drug development.
Update news regarding AOH 1996 Cancer Drug Trial Phase 1, the AOH 1996 Breakthrough in Neuroblastoma and Mouse Models. Join us on a journey through the frontier of medical innovation as we revisit AOH 1996, a groundbreaking compound at the forefront of cancer research. Previously, we've delved into the various facets of this drug's discovery and its ongoing Phase 1 clinical trial. Today we're shining a light on the insights of an expert in the field, integrating her perspectives into the narrative of this potentially revolutionary trial, with outcomes eagerly anticipated in September 2024. AOH 1996 stands as a testament to years of dedicated research and development, crafted to precisely target and curb tumor growth. As we edge closer to this pivotal moment in September 2024, let's unravel the complexities of this significant research and its potential impact on cancer treatment. In today's feature, we dive deep into the enigmatic world of AOH 1996, a small molecule ligand under the microscope for its potential in revolutionizing cancer treatment. Born from the corridors of cutting-edge research and development, AOH 1996 has embarked on a clinical odyssey, navigating through the complexities of cancer biology to offer a new beacon of hope. As we traverse the landscape of this scientific inquiry, we encounter the insights of Professor Dorothy Bennett from St. George's University of London. Her expert analysis sheds light on the findings published in Cell Chemical Biology. Professor Bennett describes the innovative approach of targeting the pCNA protein, a critical player in the dance of DNA replication and repair, a pathway where cancer cells often find their foothold. However, the path to medical breakthroughs is fraught with stringent scrutiny. Professor Bennett points out that, while AOH 1996 has demonstrated a capacity to slow down the growth of various cancer cell lines, the evidence of its ability to outright kill cancer cells remains inconclusive. In her view, the data primarily indicate a retardation of growth rather than a definitive cessation or reversal, especially in the context of human neuroblastoma lines and in vivo tumor models in mice. To make this clearer, I am including an explanation that refers to the specific experimental settings used to study the effects of a treatment, in this case, possibly AOH 1996 or a similar compound on cancer growth and progression. Let's break this down further for a clearer understanding. Human neuroblastoma lines. Neuroblastoma lines. These are cell lines derived from human neuroblastoma tumors, a type of cancer that usually arises in nerve tissue of infants and young children. These cell lines are grown and maintained in laboratory conditions to study the biology of neuroblastoma and to test potential treatments. Research purpose. By using human neuroblastoma cell lines, researchers can investigate how cancer cells respond to new drugs at a cellular level. They can observe changes in cell growth, survival, gene expression, and other cellular processes in response to treatment. In vivo tumor models in mice. In vivo models, these are live animal models, in this case mice, into which human cancer cells or tissues have been introduced to grow tumors. These models aim to replicate the complexity of how a tumor behaves in a living organism, including interactions with the surrounding environment and the immune system. Mice models. Mice are commonly used for in vivo studies due to their biological similarity to humans, rapid reproduction, and the ability to manipulate their genetic makeup. This makes them valuable for studying disease mechanisms, testing drug efficacy, and observing the potential side effects of treatments. Tumor growth and response. In these models, researchers can monitor how well a new drug like AOH1996 inhibits tumor growth, spreads, or influences survival. The effects on the tumor can be directly observed and measured over time, providing insights into the therapeutic potential of the drug. Combining both approaches. Using both human neuroblastoma lines and in vivo tumor models in mice allows researchers to gain a comprehensive understanding of a drug's efficacy. In vitro studies with cell lines provide initial insights into the drug's mechanism of action and potential against cancer cells. Subsequently, in vivo studies offer a more complex picture of how the drug performs in an organism, taking into account factors like metabolism, immune response, and drug distribution. In summary, studying the effects of new cancer treatments in the context of human neuroblastoma lines and in vivo tumor models in mice enables a thorough investigation from cellular-level responses to whole-body efficacy, paving the way for advancing these treatments to clinical trials in humans if proven successful. Despite these reservations, the silver lining emerges in the broad-spectrum efficacy of AOH1996 against multiple cancer types, coupled with its minimal impact on normal cell types. This dichotomy fuels the argument for further development, suggesting that the journey of AOH1996 is far from over and deserves continued exploration. As we look towards September 2024, the anticipation grows for the culmination of years of research and development into AOH1996. The upcoming results promise to either elevate this molecule to the next phase of clinical trials or beckon a return to the drawing board. In this ever-evolving narrative of medical science, every data point, every trial, and every expert opinion like that of Professor Bennett plays a crucial role in shaping the future of cancer therapy. This video aims to encapsulate the ongoing research on AOH1996, integrating expert critique with the anticipation of forthcoming trial results to provide a comprehensive and engaging overview for the audience. Stay tuned as we continue to monitor the progress of AOH1996, bringing you the latest updates and expert perspectives. Your engagement with our content not only fuels our journey, but also lights the way in the quest for knowledge and the hope for a cure. Don't forget to like, share, and subscribe, ensuring you're always connected with the forefront of medical research.